Imagine you were tasked with building a zoo, right? But there is a massive catch.
Uh-oh, I feel like there's always a catch in these scenarios.
Oh, definitely. So the catch is you can't actually see any of the animals you were trying to house.
Wait, you can't see them at all?
Not at all. You have zero idea if they're predators or prey, if they live in trees or, underground, or what they even eat.
Okay, that is a nightmare for a zookeeper.
Right. So you basically just throw a stake and maybe a pile of leaves into a concrete box. You lock the door and you just hope something survives. It sounds completely absurd.
I mean, yeah, it sounds like a recipe for a very empty, very tragic zoo, honestly.
Exactly. Well, welcome to our deep dive on culture media. Today, we are unpacking a fascinating set of documents and research notes all about the science of how we feed microbes in the lab.
Yeah, and for over a century, that invisible zoo scenario you just described, that is basically how scientists had to study the microscopic world all around us.
It really is.
Yeah.
So whether you are cramming for a biology meeting tomorrow, catching up on microbiology, or you're just fiercely curious about how scientists coax invisible life into plain sight, this deep dive is your ultimate shortcut.
Absolutely.
We're going to explore how we figured out what to feed these invisible beasts, the kitchen trick that changed medicine forever, and the really humbling realization that we might still be getting it completely wrong.
It really is a fascinating journey because trying to keep a microbe alive outside of its natural environment is, well, it's an immense engineering challenge.
Oh, I can imagine.
Yeah, you are basically trying to simulate an entire ecosystem in a little plastic dish. You're manipulating chemistry to mimic nature.
Okay, let's unpack this. What exactly is a culture medium?
Right, the core definition.
Yeah, simply put, it is an engineered nutrient preparation. It can be solid, it can be liquid, or semi-solid. And it's specifically formulated to support the growth of microorganisms in a controlled laboratory setting.
So we are not just tossing a cracker in a jar here.
No, definitely not. At a bare minimum, a viable medium has to supply a carbon source, a nitrogen source, water, and essential minerals like sulfur and phosphorus.
Right, the basic building blocks of life.
Exactly. Plus, for a lot of organisms, you need specific growth factors, like vitamins or amino acids, that the microbe simply cannot manufacture for itself.
And if you look at the history, the very concept of what it means to actually grow a microbe has quietly shifted over the last 150 years or so.
Really? How so?
Well, in the earliest days of bacteriology, growing a microbe just meant you had a beef broth that eventually turned cloudy.
Just cloudy water.
Right, cloudiness equaled life. But by the 1880s, the standard became much stricter. It meant watching a visible, isolated colony form on a solid surface.
Ah, so you could actually see a little dot of bacteria.
Exactly. That visual proof allowed scientists to claim they had a pure culture of a single species.
And reading through these sources, that definition has splintered even more today. Like, microbiologists have had to face a pretty hard truth, which is that most microbes on Earth absolutely refuse to grow on anything humans have devised.
Yeah, they are extremely stubborn. Modern textbooks actually categorize this now.
Right. You have organisms that are culturable, meaning that they will grow on some known medium. Then you have fastidious organisms.
Which is such a great word. Fastidious.
It is. They're incredibly picky and need highly complex, nutrient-rich media. And then you have organisms in what is called a VDNC state.
Which stands for viable but non-culturable, right?
Exactly. Viable but non-culturable. They are alive, they are metabolically active, but they will not form a colony under any lab condition we have ever tested.
Wow.
So a culture medium is no longer just a recipe. It's really a statement about the limits of what we can coax life to reveal itself.
That is so true. And to truly appreciate how we feed microbes today, we have to look back at the messy, melted disasters scientists were dealing with in the 1800s.
Oh yeah, the early days were rough.
Very rough. This whole field grew directly out of the germ theory revolution. In the 1860s, Louis Pasteur was using liquid nutrient broths, basically sugar and yeast solution.
Right, to disprove spontaneous generation and study fermentation.
Exactly. But these liquid media had a massive critical flaw for studying specific diseases.
Think of Pasteur's liquid broths, like a blended smoothie. If you drop an environmental or clinical sample into a liquid, every single species in there just swirls together.
Yeah, it's a total free-for-all.
You can never isolate just one fruit to study it, so to speak. You just have a chaotic smoothie of several different organisms reproducing all at once.
Which is useless if you're trying to figure out what's causing a disease.
Exactly. If you were trying to prove which specific microbe causes tuberculosis or cholera, a mixed smoothie doesn't help you at all.
Which brings us to Robert Koch. Working in Germany in the early 1880s, Koch desperately needed to isolate pure cultures.
Because of his postulates, right?
Right. He was developing what we now call Koch's postulates. They're the gold standard rules for proving a specific microbe causes a specific disease.
Gotcha.
And the core requirement of those postulates is that the suspected pathogen must be isolated in a pure culture. You have to prove it is that one specific bacteria and nothing else making the host sick.
So you need to separate them out.
Yes. Solid media is the literal instrument that makes Koch's postulates possible. Without it, you cannot establish rigorous causal proof.
And you couldn't do any of this without developing aseptic technique at the same time, right?
Oh, absolutely not.
Like flame sterilizing tools, sealing containers. Because if you create the perfect microscopic buffet, every mold spore floating in the room is going to want a seat at the table.
Yeah, they will crash the party instantly.
If you don't keep contaminants out, your pure culture is completely ruined before you even start.
So Koch knew the liquid smoothie wasn't going to cut it. He needed a solid surface. And his first attempt was surprisingly rustic.
Oh, the potato.
Yes, the potato. He literally sliced boiled potatoes and let bacteria grow on the moist cut surface.
I just love the image of a world-class scientist staring intensely at a moldy potato.
It's very glamorous science, right?
So glamorous. But the sources note it was far too crude. I mean, you couldn't standardize a potato, obviously.
Right. Every potato is different.
And it certainly wasn't transparent enough to inspect the bacterial colonies closely. So Koch moved to his next idea, which was gelatin mixed with nutrient broth, poured onto glass plates.
Which sounds like a good idea in theory.
Yeah, the gelatin solidified, and if you spread the bacterial cells thinly enough, each individual cell would grow into a separate, physically isolated colony. Visible, undeniable proof of a pure strain.
But gelatin introduced two absolute disasters.
Rough.
First off, it melts at human body temperature, which is around 37 degrees Celsius.
Oh, right.
And Koch was studying human pathogens.
Yeah.
To get them to grow, you absolutely have to incubate them at 37 degrees.
So his beautiful solid plates were just melting back into muddy petals in the incubator.
Exactly, just reverting back to the chaotic smoothie.
And the second problem is purely biological. Many bacteria produce enzymes called gelatinases that actually digest gelatin.
Yeah, they literally eat the floor they're standing on.
Right. Even if you kept the room cool, the bacteria would liquefy the whole plate.
Yeah.
So Koch was just stuck.
And this is where the hero of modern microbiology steps in. And the best part is, she wasn't even a microbiologist.
Fanny Hess?
Yes, Fanny Hess. She was an illustrator for the lab and the wife of Koch's associate, Walter Hess. She sees them struggling with this melting gelatin, and she suggests they try something she uses in her own kitchen.
Agar.
Exactly. It's a polysaccharide extract from red algae that she used to set her jams and jellies.
And she learned about it from a Dutch acquaintance, right?
Yeah. Right, someone who had lived in Java, where agar was a traditional Southeast Asian food thickening agent.
What's fascinating here is the sheer physical brilliance of agar for this specific scientific problem. Like, it is practically magic.
It really is perfect for the job.
Agar only melts when you heat it to near 100 degrees Celsius, which is boiling. But once it melts, it stays in a liquid state until it cools down to below roughly 40 degrees Celsius.
Right, so you can melt it, let it cool enough to safely mix in your delicate nutrients or even living cells without boiling them alive.
And then pour it into a dish where it's set solid.
Yes, and it won't melt again even at that crucial 37-degree human body temperature.
Okay, I have to jump in and bust a massive misconception here because it trips up so many people learning about this.
Oh, go for it.
Agar itself is not the food. It contributes zero nutritional value to the microbes.
That is such an important distinction.
Right. Almost no bacteria possess the enzymes to digest it. It is purely the structural gelling agent. It is the plate, not the meal. All the actual food, the broth, the blood, the extracts, has to be dissolved into the agar.
Exactly. Koch adopted this in his lab around 1882, and honestly, it remains the absolute backbone of solid culture media to this day.
It's amazing that a kitchen trick is still the standard.
It really is. And to complete the physical setup, we have to mention Koch's assistant, Julius Richard Petri.
Ah, the Petri dish.
Yes. In 1887, Petri invented the shallow, stackable, lidded glass dish. That seemingly simple invention solved the practical nightmare of pouring, storing, and incubating these agar plates without dust from the air contaminating everything.
So Koch solved the structural problem. He had a plate that wouldn't melt and a dish to keep it sterile. But like we said, a plate isn't a meal.
Right.
If you want a specific pathogen to reveal itself out of a mixed clinical sample, you have to engineer the nutrients to cater specifically to that organism's biology.
Which brings us to the core concepts of media design.
Yeah, I'll get into the menu.
So depending on what you want to do, you change the physical state or the nutritional complexity. If you want to grow a massive army of cells really fast, you leave out the agar and use a liquid broth.
Makes sense.
If you want to see if the bacteria are modal-like, if they can actively swim, you use a semi-solid medium with just a tiny pinch of agar to make a soft gel.
Oh, so they can swim through it.
Exactly. And on the nutritional side, you have define synthetic media where you know the exact chemical quantity of every single ingredient. This is essential when you need to know precisely what a microbe requires to survive.
And on the flip side of that, you have undefined or complex media,
Right.
like Luria Bertani broth, LB broth. These use digested extracts of yeast or meat or soy. The exact chemical composition varies from batch to batch, but they offer a broad, redundant buffet of nutrients that basically supports a huge variety of species.
But here's where it gets really interesting, though. Media design isn't just about feeding things, it's about filtering them.
Filtering, right.
This is where selective and differential media come in. Selective media are designed to suppress unwanted organisms while allowing your target organism to grow.
Right. A classic example in the documents is McConkey agar. It was developed around 1900 to isolate enteric, or gut, bacteria from stool and water samples.
Yeah, McConkey is a staple.
And it contains bile salts and a dye called crystal violet. Let's look at how that filtering actually works on a structural level, because it's pretty cool.
It's basically chemical warfare.
It is. Gram-positive bacteria have a thick, exposed mesh-like layer on their outside. The crystal violet dye and the bile salts essentially get tangled in that mesh and poison them, completely disrupting their cell walls.
But the gram-negative bugs survive.
Exactly. Gram-negative gut organisms like E. coli and Salmonella, they have an extra outer membrane that acts like armor, keeping the dye out. So they just grow basically unopposed.
And you know, people often hear the word selective and think it means the plate instantly kills everything else on contact.
Oh, right, like a sterilizer.
Yeah, but it doesn't sterilize the sample. Selective media just inhibit or slow down the unwanted growth. That's why if you leave a selective plate in the incubator for way too long, those suppressed organisms can eventually fight through and start to overgrow.
And that makes total sense. Now, McConkey is selective, but it's also differential, meaning it doesn't just filter, it diagnoses.
Yes, the color coding.
Right. It contains a sugar, lactose, and a pH indicator dye. If the microbe growing on it ferments lactose, like E. coli does, its metabolic byproduct is acid.
And acid lowers the pH.
Exactly. That acid drops the pH of the agar, reacting with the indicator dye to turn the colony and the surrounding area bright pink.
It's very dramatic looking.
It is. But if it doesn't ferment lactose, like Salmonella, no acid is produced and the colonies just stay pale, you get a single plate that filters out the junk and literally color codes the pathogens for you.
It's brilliant engineering. Then you have enriched media for those fastidious, picky eaters we mentioned earlier. Blood agar is the standard here.
Blood agar. Sounds a bit morbid.
A little bit, yeah. It's a nutrient base with 5% sheep blood added. And not only does it feed demanding pathogens like Streptococcus, but it also acts as a differential medium.
How does the blood differentiate them?
It reveals hemolysis patterns. Hemolysis is the specific biological mechanism bacteria use to destroy red blood cells so they can scavenge the iron inside.
Oh, wow. So if a bacterium produces an enzyme that completely ruptures the red blood cells, you get this entirely clear halo around the colony. Right.
That's beta hemolysis.
And if it only partially breaks them down, oxidizing the hemoglobin, it leaves a dull greenish ring, which is alpha hemolysis.
Exactly. And if the blood is untouched, that's gamma hemolysis. That single visual cue is a massive shortcut for identifying different types of strep throat in a clinical lab.
Okay, which leads us to the most bizarrely named medium in all of microbiology.
Oh, I know where you're going with this.
Chocolate agar. I have to demand the truth on this one. Is there cocoa in this? Is it sweetened? Because it sounds like a dessert for bacteria.
I hate to ruin the illusion, but it contains absolutely zero chocolate.
Disappointing.
I know. It is actually just blood agar that has been gently heated. The heat lyses or bursts open the red blood cells. Cooking the blood like that turns the medium a brown chocolatey color.
So it's basically just cooked blood.
That is exactly what it is. And heating the blood is necessary for certain microbes. It releases intracellular nutrients, specifically compounds like NAD and hemin, that are securely locked inside intact red blood cells.
And some bacteria can't get to them otherwise.
Right. These nutrients are required by exquisitely fastidious pathogens like Haemophilus influenza and Neisseria gonorrhea. They are so demanding they cannot even grow on regular, unheated blood agar.
Wow.
Yeah, they lack the enzymes to burst the red blood cells themselves, so they need you to cook the blood and break the cells open for them first.
It is incredible how specific these biological requirements get. But, sometimes you actually want a recipe with almost nothing in it at all.
Right, stripping it back.
Minimal media. They contain just the bare essentials, a single carbon source, some salts, water, and 0 extra growth factors. Scientists use it to test mutant strains of bacteria or fungi.
Oh, this is such a clever use of media.
It really is.
Yeah.
If you take away everything but a basic sugar, a healthy microbe synthesizes its own vitamins and survives. But if a mutant dies on minimal media unless you manually add a specific vitamin, you found a broken biological pathway.
And that exact logic is what underpinned George Beadle and Edward Tatum's experiments back in 1941.
The Nobel Prize winners.
Yep. They worked with a bread mold called Neurospora. By comparing how the mold grew on minimal media versus supplemented media, they figured out that specific gene mutations corresponded to the loss of specific enzymes.
Wow.
It established the one gene, one enzyme hypothesis, which won them a Nobel Prize, all from just tweaking the menu on a culture plate.
That's amazing. So with all these genius recipes, from pink macaroni to heated chocolate agar to Nobel Prize-winning minimal media, it sounds like we are the absolute masters of the microscopic menu.
You would think so, right?
But reading further into the sources, there's a massive conceptual roadblock. What if all these recipes, all this brilliant engineering, are actually representing a massive failure?
Yeah, that is arguably the most consequential debate in this entire field. It is known as the Great Plate Count Anomaly.
Right. When I read about Rita Colwell's work in the 80s, coining the Great Plate Count Anomaly, it completely upended this for me.
It's a huge paradigm shift.
The anomaly is this. If you take a liter of seawater and you look at it under a microscope, you can literally count the cells. Let's say you see 1 million cells.
Okay.
But when you take that exact same sample and plate it on standard laboratory media, you might only get 1,000 visible colonies growing. You routinely see 100 to 1,000 times more cells under the microscope than ever show up on your plates.
It's staggering. Well over 99% of the microorganisms in typical environments have never, ever been successfully cultured on any medium we have invented.
That's wild. Culturability is not the rule. It is a rare exception.
It's like throwing a lavish all-you-can-eat buffet, right?
Yeah.
But 99% of the guests are extreme introverts.
I love this analogy. They don't want the buffet. They just want a quiet corner, one very specific type of cracker, and to sit next to their one best friend. And if you force them into the feast, they just shut down and refuse to eat entirely.
That is remarkably accurate. And for the first century of microbiology, every culture-based estimate of microbial diversity was essentially studying that 1% of extroverts.
Thinking they represented the whole room.
Exactly. We were working from a tiny, non-random slice of the actual microbial world.
So why do they refuse the food?
Well, the reasons come down to biology. Your introvert analogy hits on one of them. They need their friends. Many microbes require signaling molecules or metabolic byproducts from neighboring species that a pure culture plate, by definition, strips away.
Oh, because Koch's whole thing was isolating them.
Right. But we are actually starting to solve that with co-culture techniques where we let different species share chemical signals.
That makes sense. What else?
Another reason is simply time. Some organisms grow so incredibly slowly that before they can form a visible colony, faster competitors have already overrun the plate and eaten everything.
Okay, the third reason really surprised me because it busts another major misconception. We just assume that more nutrients always means better, faster growth. Like if a microbe is struggling, just give it more food.
Yeah, human logic doesn't always apply to microbes. Standard media are often far too nutrient-rich.
Food?
Yeah. Many environmental microbes are what we call oligotrophic, meaning they are adapted to extremely low nutrient environments. So when an organism adapted to starvation hits a nutrient buffet, the sudden influx of organic material throws its metabolism into overdrive.
It's like a shock to the system.
Exactly. The rapid metabolism generates toxic levels of oxidative stress, essentially. Free radicals tear the cell apart from the inside. The metabolic shock forces it into dormancy or just outright kills it.
Wow, we're literally killing them with kindness. And this blind spot had real historical consequences for public health, didn't it? Back in the early 20th century, public health labs would test municipal water or dairy milk using a small set of standard media.
Right.
They would plate the water, count very few colonies, and just certify the water as safe.
They mistook the limitations of their tools for a clean bill of health. Because they couldn't grow the pathogens, they just assumed the pathogens weren't there.
That is terrifying.
Yeah.
Because we were essentially blind to 99% of the microbial world, scientists eventually had to launch a modern rebellion to rethink how we feed these organisms. Which brings us to why this all matters for the world we live in today.
Right. That rebellion is known as the Culturomics movement. It really took off in the 2000s and 2010s, pioneered largely by Didier Raoult's group in Marseille.
Culturomics.
Yeah. Instead of playing it safe and using one or two standard media, they essentially brute forced the problem. They took human gut samples and subjected them to dozens of different media, drastically different temperatures, varying atmospheric conditions, like stripping out all the oxygen and extended incubation times for months, all in parallel.
They just threw every conceivable environmental variable at the wall to see what stuck.
Exactly. And it worked. They successfully cultured hundreds of bacterial species from the human gut that had previously only ever been known about because we found their DNA sequences.
Wow.
Some of these species had been written off as permanently unculturable. Culturomics proved that a huge chunk of that 99% gap was just a failure of human imagination and how we designed our media, not an impenetrable biological wall.
So what does this all mean for you, the listener? Culture media secretly underpins your daily safety. Like, if you go to a clinic for a suspected UTI or strep throat, the lab is swabbing your sample across selective and differential plates.
Every single day.
Right. Within a day or two, because of things like McConkey or blood agar, your doctor knows exactly which pathogen is causing the issue, what its biology is, and exactly which antibiotic will save you.
It really is the invisible shield protecting your grocery store, too. Food safety testing relies heavily on selective enrichment media.
Oh, like checking for E. coli.
Yeah, or listeria, or salmonella. If there is a tiny trace of dangerous salmonella hiding among billions of harmless cells on a piece of lettuce, selective media mathematically filters out the harmless noise and allows the dangerous cells to multiply until we can actually detect them.
That is so crucial.
Water utilities use these media every day to test for fecal coliforms. Pharmaceutical companies rely on them to certify that vaccines are perfectly sterile. It's everywhere.
And if we connect this to the bigger picture, the push to finally culture the unculturable is fundamentally reshaping medicine. For years, we could sequence the DNA in the human gut and see that thousands of species live there. But sequencing DNA is just observing from a distance.
Exactly. But because of culturomics, researchers can now actually grow them. When you have a pure culture, you can run direct experiments to understand the mechanisms of how these specific gut microbes link to obesity, to autoimmune diseases, and even to mental health.
Right, you move from just knowing they exist to actually understanding what they do.
It's a huge leap forward.
We have journeyed from Fanny Hess realizing her kitchen jam trick could save Robert Koch's melting gelatin to the brilliant structural diagnostic power of McConkey agar, all the way to the humbling realization that 99% of microscopic life still refuses to eat our lab food.
Which, this raises an important question, something for you to really mull over as we wrap up.
Lay it on us.
If over 99% of microscopic life is essentially invisible to our best artificial environments, like if they simply refuse to survive when we try to put them in a box, what does that say about our attempts to simulate or manage ecosystems on a macro scale? We talk about geoengineering our climate or trying to build artificial biospheres to live on other planets. But if we are trying to manage a planetary buffet while remaining totally blind to what 99% of the foundational guests actually need to survive, are we just repeating the Great Plate Count anomaly on a cosmic scale?
That is a staggering thought. If you can't feed the invisible zoo, you probably shouldn't try building the planetary one just yet.
Very true.
Well, thank you for joining us on this deep dive. Keep questioning the invisible world around you, and we will catch you next time.